Next up, yes, we have Marshall Dalton, who is a postdoc at Neuroscience Research Australia. And he will be talking about mapping anatomical connectivity between the hippocampus and cortical mantle using in vivo quantitative fiber tracking.
Okay, thank you for the introduction. Thank you all for coming, and thank you to the organizers for accepting our symposium. Okay, so I'm going to just briefly touch on some work that I've been doing with collaborators at the University of Sydney here in Australia, where we've been trying to develop new approaches to map anatomical connectivity of the in vivo human hippocampus using diffusion imaging approaches. And I have no disclosures. So just as a background, we know from decades worth of tract tracing work across species that there are very complex anatomical circuits connecting the hippocampus with other parts of the brain. For those unfamiliar with these tract tracing approaches, these typically involve injection of a tracer substance into a particular part of the brain, in this case a subregion of the entorhinal cortex. These are taken up by neurons in that region, move along the axons and infect neurons that are directly connecting with this region. Then post-mortem, we can stain this to look at precisely which neurons are preferentially connecting with this injection point. And these methods have been used extensively to create these very complex maps of circuitry across species in terms of cortical connections, subcortical, and also cortico-hippocampal connections. From this work, we know that the entorhinal cortex is the primary interface between the hippocampus and the rest of the brain. The vast majority of input to the hippocampus comes through the entorhinal cortex. But we know also that it's not the only pathway, and this is often overlooked, particularly in a lot of neuroimaging work, that there are direct connections with multiple brain regions with different subregions within the hippocampus. So just as an example here, in the macaque brain, we have different colors representing different injection sites across the brain, and each of these are associated with specific patterns of projection, both along the anterior-posterior axis of the hippocampus and also its medial-lateral extent, and connecting with specific subregions within the hippocampus. Now, we know a great deal about these connections across animal models, experimental animal models, rodents, and non-human primates, but we know much less about these connections in the human brain because we can't conduct these invasive approaches, obviously, in living humans. And in relation to the evolutionary expansion of the cortex, we will expect that there are evolutionarily conserved patterns of connections, but there may also be human-specific patterns that underpin human-specific cognitive processes. Now, one way to investigate these white matter pathways in the living human brain is using diffusion-weighted imaging approaches, but conventional approaches haven't allowed us to really track into complex structures like the hippocampus. We've been limited to look at connections that are terminated at the interface, the grey matter, white matter interface of the hippocampus. So we can see connection, these very coarse estimates of connectivity, but we've got a black hole within the hippocampus where we don't know what's happening here. And that's a very clear contrast to the very detailed patterns that we can derive from tract tracing. So we wanted to address this clear gap by developing a new approach where we've leveraged recent advances in diffusion imaging analysis methods, and we developed a bespoke approach that allows us to track the white matter pathways into the hippocampus and identify their termination points with a high degree of spatial precision. Now, of course, this is not at the level of individual neurons. It's still a coarse measure, but despite this, we think that this method really represents a significant advance in the level of spatial precision that we can achieve in mapping these connections within the in vivo human hippocampus. I'm going to briefly talk about how we've applied this approach to look at different connectivity patterns. So in study one, we've mapped the connections between the hippocampus and the cortical mantle. And in study 2, we've looked more specifically at connections between the hippocampus and the entorhinal cortex, all in the in vivo brain. So first, we leveraged data, both structural and diffusion data, from the Human Connectome project. I'm not going to go into detail of the methodology here. I'm going to keep this very conceptual in nature. But if anybody has questions about the details, please come and ask me afterwards, or take a look at our paper that was published a couple of years ago describing this in eLife. But just very briefly, we can use the diffusion data to create these probabilistic maps of white matter pathways in the in vivo brain. This is an example of a 3D rendering of a whole brain tractogram representing all of the white matter pathways. And we've developed a bespoke approach where we first manually delineate the hippocampus to ensure anatomical accuracy. And this is a 3D rendering of the hippocampus here. So I'll be displaying some results on top of this 3D rendering. So if you see this later, this is what that represents. And our approach allowed us to track streamlines into the hippocampus and isolate only those pathways that are directly connecting the hippocampus with the cortical mantle. We then use the Human Connectome Project multimodal parcellation to split the cortex into 180 different subregions, and using a combination of track density imaging and an approach called SIFT2, we can quantitatively assess the density of connections between the hippocampus and every other parcel in the HCP-MMP. And when we do this, we have here highlighted the 20 most highly connected cortical regions from our analysis. It's not the only areas that are connected, but this is just the top 20 with the highest density of connections. So we can see these are primarily in lateral and medial temporal cortices, and also in occipital regions. We then conducted a more comprehensive analysis looking at how each of these regions are preferentially connecting with the head, body, and tail of the hippocampus. And we can do this at both the individual level and the group level. At the group level, we create these endpoint density maps that show the location and the density of the streamlines, the white matter pathway endpoints within the hippocampus that are associated with each cortical area. So here we just represent these lateral temporal areas seem to be preferentially connecting with lateral portions of the hippocampus, aligning with the CA1 and proximal subiculum. And also, interestingly, there seems to be a circumscribed high-density connection in the body of the hippocampus, aligning with the distal subiculum and presubiculum. In contrast, we see very different patterns of connectivity with the hippocampus between the medial, parietal, and occipital that are preferentially connecting with these posterior medial portions, aligning with subiculum and presubiculum. We didn't go into this blind. We had very specific predictions based on the non-human primate track tracing data. Just to give one example, in the macaque, injection in the retrosplenial cortex results in staining of this posterior medial portion of the hippocampus. And when we isolate the streamlines and endpoints related to retrosplenial in the human, we find exactly the same pattern here. And we had consistent results across multiple cortical areas. So it's nice that we see these evolutionarily conserved patterns of connectivity in the human, but we also found some differences. So here in TE2A, for example, in the macaque, we don't see staining in this area of CA1, whereas we did find staining here. So we do find some differences. Whether these are related to technical limitations of our approach or whether this is actual human-specific patterns of connectivity is something that we're currently actively working on. So if you're interested, please take a look at our paper we published a couple of years ago. But we think that this method overall represents a significant advance in the spatial precision that we can achieve in mapping hippocampal connectivity in the in vivo human brain, and it informs our understanding of the architecture of hippocampal-dependent memory systems in the human brain. Now, since publication of that paper, we've further refined the methods for conducting these detailed equivalent of track tracing in the human brain, to look more specifically at connections between the entorhinal cortex and hippocampus. And again here, we used a higher resolution data set and manually segmented the hippocampus and the entorhinal cortex, again for anatomical accuracy. We also looked at perirhinal cortex, but I won't be sharing any of that data today. This is currently work that's ongoing. So again, here we have a 3D rendering of these structures, and we generate and isolate white matter pathways that are directly connecting the hippocampus and the entorhinal cortex. And then we can generate the endpoint density maps showing precisely where within each structure the endpoint of each streamline is. And if we look at this in the sagittal plane, we can see these really beautiful examples of streamlines connecting the hippocampus and entorhinal cortex, and these really beautiful innovations that are coming into specific parts of the hippocampus here. And in the coronal plane, again, we see that there are particular regions where these perforations are coming through. And if we remove the noise of the streamlines, we can see endpoint density maps, these beautiful clusters of high dense connectivity along the anterior-posterior axis. And we can make inferences about where these anatomical hotspots are connecting in relation to subfields within the hippocampus. So we can analyze these endpoint density maps in a number of different ways. So here, just to give one example, we can make very specific predictions. So again, from the non-human primate literature, we know that anterior portions of the hippocampus preferentially connect with medial aspects of the entorhinal cortex, and posterior hippocampus preferentially connects with these lateral entorhinal. So to test this, we isolate specific clusters within the hippocampus. So in green here, anterior subiculum, and in purple, posterior subiculum in the tail. And we can then isolate the streamlines that are directly connecting each of these clusters with the entorhinal cortex and visualize the endpoints. And we see this is just a schematic representation. It's difficult to show these results on the actual entorhinal cortex mask. But we see very clear patterns where these anterior clusters preferentially connect with medial entorhinal and posterior with lateral. Again, very nicely aligning with predictions based on the non-human primate track tracing work. Very nicely, this is it at the level of individual participants, and we get consistent results across our participants as well, which is very nice. But we can analyze, again, this in a number of different ways. So rather than looking at individual clusters, we can look at all clusters in the hippocampal head versus those in the hippocampal tail. And again, just presenting this in a slightly different way, we see these beautiful gradients of connectivity along the medial lateral associated with the anterior and posterior hippocampus. Again, aligning with what we predict from not only non-human primate, but also in rodent literature as well. So yeah, we're really excited by this approach, and we think it really enhances the spatial precision that we can achieve in mapping these connections in the human brain. And we're currently taking this in a number of different directions. So we are looking at a method called track-weighted dynamic functional connectivity, where we combine the anatomical connectivity data with functional connectivity data, integrated so that we can look at relationships between structure and function. We are looking across an aging cohort where we're looking to use endpoint density mapping to see if we can identify specific pathways that reduce in density in the context of healthy aging to identify potentially vulnerable pathways. And also we're applying this to high-resolution data sets across species to further validate the methodology and bring new insights also into some of the track tracing work. Thank you to my collaborators in Sydney, and thank you all for your attention.